Fruit flies come in all kinds of eye colors: brick red, bright red, brown, and even pure white. But these colors aren't simple pigments. They're the outcome of interacting genetic pathways that act almost exactly like mixing paints. Today I'll use this paint analogy to explain how mutations like Harvard, Princeton, and white shape eye color through gene interaction and epistasis. Drosophila eye color is produced by two independent pigment pathways. The petadine pathway makes bright red pigment and the omicron pathway makes brown pigment. When both pigments are present, the eye color looks brick red, just like mixing these two paints. If one pigment is missing, the eye color shows the color of the remaining pathway, and if both are missing, it shows up as white eyes. Two classic mutations help us study these pathways: Harvard and Princeton. The Harvard mutation blocks the omicron pathway, so the fly loses brown pigment and keeps only bright red. In paint terms, Harvard puts a lid on the brown paint can. The Princeton mutation blocks the petadine pathway, removing red pigment and leaving only brown, so Princeton puts a lid on the red paint can. When a fly has both mutations, both pathways are shut down, neither paint can open, and the result is white eyes, because no pigment is produced at all. But there's another gene upstream of everything: the white gene, located on the X chromosome. The white gene encodes half of the heterodymic ABC transporter, a membrane pump that moves pigment precursors into developing eye colors. White can't function alone. It must pair with scarlet to import precursors for brown omicron pigment, and brown to import precursors for red petadine pigment. Think of white as the motor to the pump, and scarlet or brown as the cargo selector, which provides the ATP-driven energy. Scarlet or brown decides which pigment precursor gets imported. So here's the critical part. If white is mutated, the transporter fails completely. Neither red nor brown precursors enter the cell, even if scarlet and brown proteins are normal. In the paint analogy, white is the main pump that pulls pigment paints into the cell. So if the pump breaks, both paint cans stay closed, meaning no pigment is ever delivered. That's why the white mutation is epistatic to both Harvard and Princeton. It shuts down the production as a whole. Here's what the actual white, scarlet, and white-brown transporters look like at the molecular level. This is an ABC transporter, an ATP binding cassette transporter. The two purple columns at the top are the transmembrane domains, which form the passageway through the pigment granule membranes. The two round shapes at the bottom are the nucleotide binding domains. This is where ATP is used to drive transport. In Drosophila, white provides one half of these NBDs, and scarlet or brown provides the other half. Together they form a functional heterodimer, just like this diagram. So scarlet plus white import brown pathway precursors into the granules. Brown plus white import red pathway precursors. So this whole system physically pumps the raw pigment precursors into the lysosome-related organelles, where pigment is synthesized. And here's the key point. If white is mutated, this entire transporter complex collapses. No ATP-driven transport, no import, no precursors, and no pigment altogether. This biochemical failure explains why white is epistatic to both Harvard and Princeton. Everything breaks upstream at that transport step. These transporters sit on lysosome-related organelles, LROs, which are the pigment granules of the eye. They don't make pigment themselves, instead they deliver precursors to the enzymes inside the granule. When white, scarlet, or brown are mutated, it's not the pigment enzymes that break, it's the delivery system. The substrates never reach the granules, so the pathways shut down at the source. When we cross homozygous Princeton and homozygous Harvard flies, something amazing happens. Each mutant blocks a different pathway. Princeton blocks red, while Harvard blocks brown. But each parent carries the working copy the other one lacks. So in the F1 progeny, these functional alleles complement each other. This is a classic genetic complementation. Each parent supplies what the other is missing, restoring both pigment pathways. So the F1 flies are double heterozygote and have a wild-type brick red eye. But when we self-cross the F1 progeny, the F2 generation falls into four phenotypes. 1. Wild type, both pathways active. 2. Princeton phenotype, brown only. 3. Harvard phenotype, red only. And white, neither pigment pathway works. Because these genes act upstream in a transport system, the F2 follows a modified 9:3:3:1 epistatic ratio, where the white class is the double mutant epistatic category.